Chapter 4. A Tour of the Cell. Cell theory states two things. First, all living things are composed of cells. And two, all cells come from earlier cells. In other words, the cell theory is a theory that all living things are composed of one or more cells. Cells are the fundamental unit of life, and that which originated from pre-existing cells. Cells can be divided into two basic types: prokaryotic cells and eukaryotic cells. Biologists classify all life into three major groups called domains. These are archaea, bacteria, and eukarya. Organisms of the domains bacteria and archaea are composed entirely of prokaryotic organisms. Sometimes these are referred to as prokaryotes. All bacteria and archaea are single-celled organisms. Organisms of the domain eukarya are composed of eukaryotic cells. These are called eukaryotes. Some eukarya, such as yeast, are single-celled, but most are multicellular. Prokaryotic and eukaryotic cells have some features in common, including the plasma membrane, the cytosol, chromosomes, and ribosomes. The plasma membrane, or cell membrane, separates the outside of the cell from the inside of the cell. The cytosol is the fluid that exists inside the cell but outside of the nucleus. Both prokaryotic and eukaryotic cells have genetic information. This exists as one or more chromosomes and carry genes made up of DNA. Both prokaryotic and eukaryotic cells have ribosomes, which are the sites of protein synthesis. There are many differences between prokaryotic and eukaryotic cells. For example, prokaryotic cells have been around much longer, over 3.5 billion years ago, whereas eukaryotic cells began their existence only 1.8 billion years ago. Prokaryotic cells are also much smaller and much simpler than eukaryotic cells. Prokaryotic cells do not have membrane-bound nuclei, and they do not have membrane-bound organelles. The DNA in prokaryotic cells is located in an area called the nucleoid. On the other hand, eukaryotic cells have a membrane-bound nucleus which houses the DNA, along with membrane-bound organelles. Most prokaryotes have a cell wall. The cell wall surrounds the plasma membrane of most prokaryotic cells. This works to protect the cell and help maintain the cell shape. Some prokaryotes have a sticky outer coat called a capsule. The capsule surrounds the cell wall. It provides protection and helps it to stick to surfaces and to other cells when it exists in a colony. Prokaryotes can have short projections called fimbria. Fimbria are used to attach to surfaces along with the capsule. Many prokaryotic cells have flagella. Flagella are long projections that help them move through their liquid environment. The key difference between prokaryotic cells and eukaryotic cells is that eukaryotic cells are partitioned or compartmentalized by membranes into organelles. These membranes allow each organelle to maintain specific chemical conditions that favor the metabolic tasks performed there. The region of the cell outside the nucleus and within the plasma membrane is called the cytoplasm. In prokaryotes, the cytoplasm refers to the interior of the cell. In eukaryotes, the cytoplasm of a eukaryotic cell consists of various organelles suspended in a liquid called the cytosol. Most organelles are found in both animal and plant cells. These include ribosomes, the endoplasmic reticulum, the plasma membrane, the Golgi apparatus, mitochondria, and the nucleus. There are also differences between plant and animal cells. For example, only plant cells have chloroplasts where photosynthesis occurs, a cell wall which provides stiffness to plant structures, and a central vacuole. The plasma membrane is also called the cell membrane. The plasma membrane and other membranes are composed mostly of phospholipids. Each phospholipid is composed of a head region and a tail region. The head has a negatively charged phosphate group, which is hydrophilic or water-loving. The two tails of fatty acids are nonpolar and hydrophobic or water-fearing. Phospholipids arrange themselves into a two-layer sheet called a phospholipid bilayer. The phospholipids' hydrophilic or water-loving heads are arranged to face outward. These are exposed to the aqueous solutions on both sides of the membrane. The hydrophobic or water-fearing tails are arranged inward. They mingle with one another, shielded from the water. The phospholipid bilayer of most membranes contain proteins that help regulate traffic across the membrane. The phospholipids and most of the proteins can drift about within the membrane. The membrane is a fluid mosaic because the molecules can move freely past one another. This phenomenon is called the fluid mosaic model. Plant cell walls surround the plasma membrane. They're made up of cellulose fibers, which are long chains of polysaccharides. The cell wall functions to protect the cell, to maintain its shape, and to keep cells from absorbing so much water that they burst. The cell wall also contains channels that allows water and other small molecules to move between cells. The nuclear envelope is a double membrane that separates the nucleus from the cytoplasm. The nuclear envelope is composed of a phospholipid bilayer with associated proteins. It also contains pores that allow certain materials to pass through. The nucleus contains the DNA of the cell. DNA molecules and associated proteins form fibers called chromatin. Each long chromatin fiber constitutes one chromosome. The number of chromosomes in a cell depends on the species. For example, the human body cell has 46 chromosomes. Rice cells have 24 chromosomes, and dogs have 78 chromosomes. The nucleolus is a prominent structure located within the nucleus. The nucleolus is the site where the components of the ribosomes are made. Ribosomes are sites of protein synthesis. In eukaryotic cells, the components of ribosomes are made in the nucleolus within the nucleus. The ribosomes are then assembled from the components in the nucleus. After assembly, the ribosomes are then transported through the pores of the nuclear envelope into the cytoplasm. Some ribosomes will be suspended in the cytosol. These are called free ribosomes. These ribosomes make water-soluble proteins that remain within the cytoplasm. Other ribosomes will be attached to the outside of the nucleus, or attached to the endoplasmic reticulum. These are considered bound ribosomes. Bound ribosomes make lipid-soluble proteins that will be incorporated into membranes or secreted by the cell. The sequence of events during protein production in a eukaryotic cell are as follows. Step 1. DNA's coded information gets transcribed into a molecule called messenger RNA, or mRNA. Step 2. mRNA exits the nucleus and binds to a ribosome. Step 3. The ribosome moves along the mRNA, translating the genetic message into a protein with a specific amino acid sequence. The endoplasmic reticulum is connected to the nuclear envelope. It forms an extensive labyrinth of tubes and sacs running throughout the cytoplasm. There are two components that make up the endoplasmic reticulum: the rough endoplasmic reticulum, or rough ER, and the smooth endoplasmic reticulum, or smooth ER. The rough ER contains ribosomes, making it appear rough. The rough ER manufactures and packages secretory proteins. The smooth ER contains no ribosomes, making its appearance smooth. The smooth ER synthesizes lipids. The Golgi apparatus receives, refines, stores, and distributes proteins. Proteins from the rough ER are transported to the Golgi apparatus in transport vesicles. One side of the Golgi stack serves as a receiving dock for vesicles from the ER. Proteins are then modified, and the finished products are then carried in transport vesicles to other organelles or to the plasma membrane. A lysosome is a membrane-enclosed sac of digestive enzymes. This functions to break down substances using digestive enzymes. Lysosomes function to access nutrition, rid of cellular debris, and to recycle cellular components. Many single-celled protists engulf nutrients into tiny cytoplasmic sacs called food vacuoles. Lysosomes fuse with the vacuoles, exposing the food to digestive enzymes. Small molecules that result from this digestion, such as amino acids, then leave the lysosome to nourish the cell. Lysosomes can also help destroy harmful bacteria. For example, your white blood cells ingest bacteria into vacuoles. The lysosomal enzymes then rupture the bacterial cell walls, destroying the invaders. A lysosome can engulf and digest parts of other organelles, recycling it and making its molecules available for the construction of new organelles. Vacuoles are large vesicles with a variety of functions. There are food vacuoles, contractile vacuoles, and central vacuoles. Certain freshwater protists have contractile vacuoles. These pump out excess water that flows into the cell. A central vacuole is found in plants. This stores organic nutrients such as proteins stockpiled in the vacuole of seed cells. The central vacuole contributes to plant growth by absorbing water and causing cells to expand. In the cells of flowering petals, central vacuoles may contain pigments that attract pollinating insects. Central vacuoles may also contain poisons that protect the plant against plant-eating animals. Photosynthesis is the conversion of light energy from the sun to chemical energy of sugar molecules. Chloroplasts are the photosynthetic organelles of plants and algae. Chloroplasts have their own DNA. This DNA is in the form of a single circular chromosome that resembles a prokaryotic chromosome. Chloroplasts reproduce independently of the cell. They are believed to have evolved from an ancient free-living prokaryote that established residence within other larger host prokaryotes. This is called the endosymbiotic theory. Endosymbiotic theory theorizes how chloroplasts and mitochondria have come about in eukaryotic cells. Endosymbiotic theory states that a prokaryote ingested some aerobic bacteria in the past. These aerobes were protected by the prokaryote and produced energy for the cell. Over a long period of time, these aerobes became mitochondria, and they lost their ability to live on their own. Some of these primitive prokaryotes also ingested cyanobacteria. Cyanobacteria contain photosynthetic pigments. Over a long period of time, these cyanobacteria in the prokaryotes became chloroplasts and could not live on their own anymore. A chloroplast is divided into compartments by membranes. The innermost compartment holds a thick fluid called stroma, which contains DNA, ribosomes, and enzymes. A network of sacs called thylakoids is suspended within the stroma. The stacked sacs are called granum, or grana for plural. These grana are solar power packs converting light energy to chemical energy. Mitochondria are the organelles in which cellular respiration takes place. During cellular respiration, energy is harvested from sugars and transformed into another form of energy called ATP, or adenosine triphosphate. Cells use the chemical energy of ATP as a direct energy source. Mitochondria are found in almost all eukaryotic cells, including those of both plants and animals. A mitochondrion is enclosed by two membranes separated by a narrow space. The inner membrane encloses a thick fluid called the mitochondrial matrix. This inner membrane has many enfoldings called cristae, which increase the surface area, maximizing ATP output. Like chloroplasts, mitochondria have their own DNA that is in a form of a circular chromosome that resembles prokaryotic chromosomes. They also reproduce independently of the cell. The cytoskeleton is a network of protein fibers extending throughout the cytoplasm. These allow for support, maintain the cell shape, and provide anchorage for many of the organelles. The cytoskeleton also allows for movement. For example, it allows for the crawling movement of the protist amoeba, and allows for the movement of some of our white blood cells. The cytoskeleton contains several types of fibers: microtubules, intermediate filaments, and microfilaments. One function of the cytoskeleton is to give mechanical support to the cell and to maintain its shape. This is especially important for animal cells, which lack the rigid cell wall. Flagella are located in some eukaryotic cells. They are long extensions from the cell that aid in movement. These propel cells with an undulating, whip-like motion. Human sperm cells have a single flagellum. Some protists have one flagellum or many flagella. Cilia, singular cilium, are found in some eukaryotic cells. These are short extensions from the cell that aid in movement. They move in a coordinated back and forth motion that propels various protists through the water. Thank you for watching.